All right, so for this next lecture, we are going to talk about enzyme classes. So what is an enzyme and where do they come from? Well, an enzyme is basically something that will speed up a chemical reaction. And for us, in order for life to exist, enzymes are required to get over a threshold needed for a reaction to move forward. And so enzymes are very important. And where do they come from? They come from our DNA. So enzymes are encoded in our DNA and they become proteins. So they go from DNA to RNA to proteins. And that's basically what enzymes are. So we make all of our own enzymes. Now there are classes of enzymes. We have our oxireductases, we have our transferases, and we have our hydrolases. And those constitute the most abundant enzymes we have. Hydrolases are by far the most abundant. And then oxireductases and transferases come in close seconds. And then we have ligases, isomerases, and ligases. So the purpose of this presentation is just to get you familiar with the different classes of enzymes that we are going to be discussing throughout the course. So for oxireductases, they are basically going to catalyze oxidation reduction reactions. And basically, electrons are going to be transferred from the reductant to the oxidant. So you are going to move hydrogen, for example. Hydrogens are a common form of electrons that get transferred in a chemical reaction. And so you're going to have NAD is going to pick up a hydrogen. And this is an example shown here is ethanol being with alcohol dehydrogenase as the transferring the hydrogen. And so NADH picks up the electrons. This is NADH, NAD, and NADP are referred to as cofactors, and they are often associated with these oxygen reduction reactions. These reactions are going to be found in glycolysis, TCA cycle, oxidative phosphorylation. All cells that can have mitochondria are going to use these. Cells that undergo glycolysis, which basically is every cell in your body, are going to use these types of enzymes. The next class is a transferase. And a transferase is simply what it says it is. It's going to move a functional group from one molecule to another molecule. So you're going to have AX plus B, and you transfer that X from A to B. Examples are going to be kinases, deaminases. So kinases are going to be involved in phosphates. Deaminases are going to be involved in nitrogens, and they are going to move those around. So kinase will transfer a phosphate group, and this is going to equal phosphorylation. And this is going to target lipids, carbohydrates, many other things. The transfer of phosphate groups is also important for controlling signal transduction pathways. I'm going to move this deaminase. Let me put it here. Transfer of phosphate also is involved in controlling. So when you have insulin signaling, glucagon signaling, hormone signaling, different types of signaling are going to have different transduction pathways, and phosphorylation is an easy way to control, turn on one and turn off one. And there are over 500 different types of kinases in our body. And this just shows you over here a kinase transferring a phosphate. So here we have some sort of protein, and then we're going to have a serine, threonine, or tyrosine side chain, and you're going to have different enzymes that work depending on what the side chain is. But in general, you have some sort of kinase that is going to take a phosphate from ATP, generate ADP, and transfer that phosphate to either that serine, threonine, or tyrosine side chain. And this becomes a phosphorylated protein. Now, on the other side, we're going to have phosphatases. And this is going to be able to dephosphorylate that protein, remove the phosphate from that protein. So these are different types of transferases. We also have deaminases, and this is going to transfer an amine group. So these are going to be important in breaking down amino acids after a high protein meal. And these are going to involve the generation of ammonia. And so, for example, you have adenosine, and you have an adenosine deaminase, which is going to release your ammonium ion. And this is basically going to remove that nitrogen and convert it to inosine. Now, hydrolases are the most abundant, and they're going to be able to break single bonds, and they do this by just simply adding water. And of course, our body is made up of a lot of water. Examples of this are going to be proteases, peptidases, and these can break down proteins. We also have lipases, which are going to break down lipids. And we have nucleases. And these are going to cleave the phosphodiester bonds. Between nucleotide subunits. And so here we have chymotrypsin, which is going to break apart this protein into two parts by the introduction of water. So you're going to add an oxygen to one and hydrogen to the other unit. Now, ligases are going to catalyze reactions that break chemical bonds, and after they break the chemical bonds, often a ring or a double bond is going to form. And so this is an example of isocitrate, and you have isocitrate lyase, and this is going to form succinate and glycolate. And so you can see you have a double bond on that oxygen formed. The opposite reaction of a lyase is a synthase reaction. In which you have a substrate is added. Now, carboxylases are going to add or remove carboxyl groups. Let me put an example here. They add or I should say or remove. And so carboxylases are going to be important. When we start to talk more about biochemistry, one thing I'm always going to mention is counting carbons. So some key carboxylase reactions is when you have a pyruvate, which is 3 carbons, and you go to acetaldehyde. which is a 2-carbon, you're basically removing 1 carbon. And these are not easily reversed. And so this is going to require pyruvate decarboxylase. And this requires also the coenzyme, TPP. And another that's very important is the conversion of pyruvate to oxaloacetate in the mitochondria. And this occurs with a pyruvate carboxylase. And in this case, you're going from a 3-carbon to a 4-carbon. So you're adding a carbon. And this is going to be in the presence of biotin. And we'll talk more about that. But these are 2 important reactions. This is a carboxylase, which is going to either be taking away a carbon from pyruvate to acetaldehyde, or a pyruvate carboxylase that is going to be adding a carbon from pyruvate to oxaloacetate. Now, isomerases are going to catalyze structural changes within a molecule. So this is only 1 substrate and 1 product. Nothing is gained, and nothing is lost. You're just moving things around. So we have dihydroxyacetone phosphate shown here, and this by a triose phosphate isomerase becomes glyceraldehyde 3-phosphate, and this is of course in glycolysis. And so you can see that you're just moving around your hydrogens, and so this is just causing the hydrogens to move. And most of these isomerase type reactions are because the next step, the enzyme, requires a certain conformation in order to bind to the substrate. And so you must alter the conformation so that the enzyme substrate fit. And now a ligase is going to catalyze the joining of 2 substrates together. In the case of a ligase, you usually need some potential energy. So oftentimes it's going to be coupled to the hydrolysis of a diphosphate bond of ATP. So this is bringing or catalyzing joining of 2 substrates. Requires energy. So it's coupled to okay, so this is the case we're looking at DNA ligase. So you're bringing together or ligating 2 areas of the DNA molecule. And so in this case, you're going to have ATP, which is going to donate a phosphate. And then you're going to have AMP transfer the energy from that phosphate to eventually form this bond to bring together the 2 cysteine residues. Okay, so basically here, know the different enzymes, know your hydrolase, oxireductase, lyase, transferase, ligase, and your isomerases. And that is the end.